Welcome back, everyone. So welcome to, wow, we're already in topic number 3. So in this topic, we're going to kind of switch focus. So instead of just studying kind of the chemistry of life, we're going to actually study cells, and we're going to start looking at cell structure and the function of cells in chapter 3 of our OpenStax textbook. If you are in our Campbell Biology textbook, it would be chapters 4 and 5. So we're going to start out with basically looking at how, we've been studying our living things, starting with the smallest part of our life's hierarchy, starting with the atom. And then how, we have two atoms connected together with a chemical bond forming a molecule. And then we learned about how we can unite our molecules with dehydration synthesis reactions to form macromolecules. And then now we're going to get into how macromolecules, our carbs, lipids, proteins, and nucleic acids can form the organelles of cells. Okay, so cells being the smallest unit of life. So we're going to study the structure of the cells and the function of those structures. Okay, so we're really starting to get into cellular biology. So we're going to start out this unit by looking at 3.1, which is how cells are studied. So by the end of this chapter, you should be able to describe the roles of cells in organisms. You should also be able to compare and contrast how we study cells using light microscopy and electron microscopy. And then finally, you should be able to summarize cell theory. Okay, so these, again, good idea to write these down and then be able to address them after the video as kind of just like a review, and also to be able to organize your notes a little bit better. All right, so let's start out by talking about how cells are studied. Now, we already have gone over basically in the past 2 topics how cells are the smallest unit of life. So they have all of life's properties. So we talked about that in the first unit, life's characteristics and properties. And then we talked about how we can continue life's hierarchy by talking about how cells we can have cells that of different types that can come together to make tissues. And then tissues can make organs, and then organs work together to form organ systems. And then finally we get an organism. So cells are the basic building blocks of all organisms. So cells can be the actual organism. So we can have single-celled organisms, such as bacterial cells, which are prokaryotic. So remember, we talked about this in chapter 1. How we can take every single cell in the entire world and divide it into 2 categories. They're either prokaryotic or eukaryotic. So prokaryotic cells are those small, simple cells that lack a nucleus. And the eukaryotic cells, they're the ones that have that true nucleus where the DNA is housed within. So our cells, the cell itself could be the actual organism, and if it's a prokaryotic cell, they only come as single-celled organisms. So you'll never see a multicellular prokaryote. Eukaryotes, we can find eukaryotic organisms as either being single-celled or multicellular. So we do have single-celled eukaryotes, such as like paramecium, which are a type of protist. Yeast cells, it's a type of fungus. Okay, so those are examples of single-celled eukaryotes. Or we can have multicellular eukaryotes, such as yourself. Okay, plants, mushrooms, fish, that kind of thing, are examples of multicellular eukaryotes. All right, so we can take a look at our cells being the basic building blocks of all organisms, or being the organism themselves. Now, of course, cells, they vary in size. Now, when we look at a cell, most likely we're going to need a microscope to see it. Okay, so if we take a look at this figure, it gives us an idea of the range in general range of different cells and parts of cells. Okay, so you can see here we have, and I kind of included some general ranges over here, prokaryotes. Remember, those would be like bacterians and archaeans. They range anywhere from 1 to 10 micrometers. So remember micrometers? Micro is 10 to the minus 6. Okay, so that's really, really small. So here's 1 micrometer to 10 micrometers. So this is the relative size of a bacterial cell. So anywhere from here to here. Now, if you look at this range, you can see mitochondria. Mitochondria, that is an organelle that's found inside of a eukaryotic cell. Okay, so you can right away see that bacteria are a lot smaller than our eukaryotic cells, because we have actual organelle of a eukaryotic cell that's the same size as a bacterial cell. Okay, so eukaryotic cells, they're a lot larger than our prokaryotic cells. So they range anywhere from 10 to 100 micrometers on average. So you can see here 10 to 100 micrometers for our average plant and animal cell. Now this is average, okay? So some cells, most cells you cannot see with without a microscope. So you, for example, the cells of your body you cannot see without a microscope. So you can't, take a look at your blood cell. If you're bleeding, for example, you can't see individual blood cells without a microscope. But you can see actual cells without a microscope from other organisms. So for example, a chicken egg. Okay, so a chicken egg is something that, you might see all the time if, you eat chicken eggs, eggs for breakfast. And that certainly doesn't require a microscope to see. Okay, so some of our eukaryotic cells you can see without a microscope, such as the chicken egg. The ostrich egg being the largest egg, that is definitely without, you can't even, it's probably larger than a microscope itself. I think the actual largest cell is a neuron that's found in a blue whale. I think it's about 6 meters in length. So it's really long, okay? So that's like 6 meters, that's a long neuron, okay? So that's like a school bus size. And so that's one single cell, okay? So some of our cells you can see without a microscope, but for most cells you have to use a microscope to see, okay? And then if you take a look at the scale, you can see what type of microscopes we would be able to use. So a light microscope, which is the type of microscope that you would use in a high school or a college or even in an elementary school. So those are the ones that you maybe are familiar with if you've ever used a microscope before. So you could see like a plant cell, an animal cell, maybe even bacterial cells under there. Okay. Electron microscopes, they can see really, really small objects. So if you take a look over here, lipids, proteins, what are those? Those are macromolecules. Okay, so we can get really, really small with those. Look at those in nanometers. Okay. So there are, as far as cells go, we can see them with microscopes. Anything smaller than a cell, we need a special type of microscope, electron microscope. And then as far as what a microscope is an instrument that magnifies an object. So we're going to talk about the two types of microscopes, the light microscope and the electron microscope. So let's start out with the light microscope. So the light microscope, it uses light. So that's how it uses it. So basically it has two sets of lenses that magnify the image. And so basically it has a light source, and then that light source basically bounces the light off of two mirrors, or two sets of mirrors, and then that kind of helps, provides the light to and then bounces it off those mirrors so that you can actually see the image. So when you're using, this is, by the way, it's called a compound microscope, and it has, or a compound light microscope, because it has two sets of lenses. So two sets of lenses means two sets of magnification. So you have one set of magnification coming from the eyepiece and another set coming from the objective lenses. And you'll learn more about this in your lab. So again, if you're kind of confused, it's okay. But there's like a 10 times magnification from the eyepiece, and then each of these objective lenses have their own magnification settings on there. So when we are going through these lenses, we're bouncing off of mirrors, and so because of that, the light bouncing off the mirrors, it creates an inverted image. Okay, so, and you'll see this in the lab where basically, if I was to put a image, let's say like a letter E under this microscope, if it was right side up, when I'm looking at it under a microscope, it would be inverted. So it would be an upside down E. Okay. So when I'm looking at something under a microscope, a slide here, I am being able to see it magnified. So magnification is the degree of enlargement. So most of the microscopes that you see in, like if you went to the community college, Clackamas Community College, we have microscopes that can magnify our objects anywhere from 40 to 1,000 times. Okay, so 1,000 times, that's pretty significant. Okay. So 1,000 times bigger than what you can see just looking at it with the naked eye. Now, in addition to being able to enlarge the object, the microscope also provides you with resolving power. So resolving power is the ability of the microscope to allow the eye to distinguish two adjacent structures as separate. So this is something that is important because if we are looking at an object under the microscope and it becomes too enlarged, then we suddenly can't see two things as separate. Okay, so we have to make sure that everything is focused correctly. So we have a fine focus and a coarse focus to be able to focus things so that we can see two objects as separate. Okay, so that's what this resolving power is all about. So it allows us to magnify things as well as adjust the resolving power so that we can distinguish two things as separate. All right. So this is an example of a compound microscope. Compound meaning that it has two sets of lenses. We also have at our campus, we have something called a dissecting scope, which is a little different. The compound microscope is something that you use with a slide like this. You see the little slide here, and usually what happens with this kind of thing is that you have, most likely you're using with a preserved specimen, so you have something that's, you've already taken a sample from something, it's probably like a something that's already dead or something like that. Sometimes you can stain them, so oftentimes, like in the lab, you'll see this week But in the in-person class, what we do is we actually have our students take a sample of their cheek cells. So basically, they just take a toothpick and then they gently scrape the inside of their cheeks, and then they spread the cheek cells onto a clean slide. And then because your cheek cells are pretty much transparent, it's hard to see the individual cheek cells underneath the microscope. And so what we do is we highlight the cheek cells by staining them. So we add a stain. In this case, it's called methyl blue. And that methyl blue will actually highlight the nucleus. So when we look at the cheek cells underneath the microscopes, we can actually see individual cheek cells with the nucleus stained, so that it kind of highlights the individual cells. So staining actually allows us to see individual organelles a little bit clearer, especially for cells that are more transparent. Okay, so that's something that we can do with our preserved specimens, or in this case, it's your cheek cells, so we just acquired it from you. So we can stain them to highlight certain parts. The dissecting scope is a different type of scope. I don't have a picture of it here, but what's useful with the dissecting scope, it uses light the same, just like this one, but it only has one lens. And what's useful about that one is that you can have a live specimen. So for example, if you have like a petri dish with something that's moving, okay, so that's moving around, and whereas here you have to have like a slide with a cover slip. So if you actually have like pond water that's moving around, you can have a dissecting scope and you can kind of watch it under the dissecting scope and not worry about the lens touching the water or anything. Okay, it doesn't magnify it as well. It works a little differently, but it's still a way that you can get some magnification without having to worry about any lenses touching water and getting salt or whatever on there. Okay, so there's two types of light microscopes there. All right. Now the other type of classification of microscope is the electron microscope. So these are a lot different because they use beams of electrons instead of a beam of light. And because of that, they allow for much higher magnification. So remember, if I go back, we have light microscopes. In this slide, it's showing that they can all the way go to 100 nanometers to 1 millimeter. Electron microscopes, we're going way over to 0.1 nanometers to 10 micrometers. So a lot smaller objects. Okay, we can get into maybe even atoms with an electron microscope. Okay, so we can look at higher magnifications and higher resolving power, so we can see close-ups of very, very, very small objects. The problem, though, is that since we're basically using beams of electrons, the things have to be dead. Okay, so we can't use live cells in order to look at them under the microscope. Okay, so here's an example. Here is a light microscope view. So you can see the detail of a light microscope. So this is at 25 micrometers. This is basically pretty small. This is at 1 micrometer of an electron microscope. Okay, so look at the detail. So you can see how much more detail you can get with an electron microscope. Okay. Now we have two types of electron microscopes. We have scanning electron microscopes, CEMs, or transmission electron microscopes, TEMs. So a scanning electron microscope, it scans the surface of the cell or the object that we're looking at. So it's looking at the surface. So this would be whatever the object is. So this is some sort of tissue. So it's looking at the surface of the tissue. So you can see all the different parts of the surface of the tissue. It's not going deep down inside the tissue. It's just looking at the surface. Okay. Whereas the transmission electron microscope, it actually transects it, so it goes within and transmits the cell, providing detail of the internal structure. Okay, so scanning scans the surface, transmitting, basically think like transects to find the internal structures. All right. So those are the two types of microscopes. Both of them very important in studying cells, more particularly the structures inside the cells. Okay, and then finally we're going to look at the last part of this section of the chapter, which is on cell theory. So earlier in chapter 1 we talked about theories. So scientific theories, remember, are basically broad in scope and have lots of varied evidence to support them. And so cell theory, basically, this cell theory came about quite a long time ago when scientists, they were first trying to figure out like what very, very small objects were. So particularly when they were looking at, basically the first types of microscopes, which were not really microscopes, but they were just kind of used lenses, and they were looking through lenses and they were seeing live objects that were very small. And those ancient lenses, they could see different organisms. A lot of times they were like protists, single-celled protists that they could see moving. And so later on they were able to kind of to see blocks of cells. Like think of a plant cell. Plant cells are basically just blocks of cells, and they look like little rooms. And so that's where the term cell came from, because they look like a cell, like a room. So that's these little box-like structures is what they looked like. And so this guy named Robert Hooke, he named the cell a cell. Okay. So later on we ended up with figuring out, okay, well, how did we end up with cells? And so finally the scientists, they came up with this theory called cell theory, which states that all living things are composed of one or more cells. So we know that cells are the smallest unit of life, so all living things are made of at least one cell. And the cell is the basic unit of life, okay? And then the other part of this theory is that all new cells arise from existing cells. Okay, so these components of cell theory, they were brought about in the 1800s, and they have never been proven wrong, right? So we continue to find evidence to support this every single day. So we've never seen a cell just kind of, poofing out of nowhere. And then we've never seen any living thing not composed of a cell. All right, so that's why cell theory has, kind of stood the test of time. It still exists. This cell theory has been unchanged for so long. All right, so we're going to go through a couple review questions, and then we're done with this section. So when viewing a specimen through a light microscope, scientists use blank to distinguish the individual components of cells. Is it a beam of electrons, radioactive isotopes, special stains, or high temperatures? So what do scientists use to distinguish individual components of cells? If you said C, special stains, I would agree with you. So just like with the cheek cells that are used in the in-person part of our lab, they use a special stain in order for us to be able to see. our nucleus of our cheek cells. Okay, so C is the correct answer. The blank is the basic unit of life. Is it an organism, a cell, a tissue, or an organ? If you said the cell, I would agree with you. So the cell is the basic unit of life. So we've been talking about that over and over again. All right, so that's it for the first section of chapter 3. By now you should be able to describe the roles of cells in organisms, as well as compare and contrast light microscopy and electron microscopy, as well as summarize cell theory. So for the next video, 3.2, we'll be comparing the difference between prokaryotic and eukaryotic cells.